Hello everyone. Today we will be showing you the MitoPlate protocol for analysis of eukaryotic cells, beginning with the MitoPlate S-1 for substrate metabolism analysis and followed by the MitoPlate I-1 for inhibition analysis. The MitoPlate protocol is designed to generate kinetic data on the metabolism of mitochondria from virtually any eukaryotic cell. Today we will be testing a typical colon cancer cell line, Colo 205, on both plate types. From here, I will let Kyle O'Halloran, one of Biolog's most experienced end users of the MitoPlate, show you how each plate is run, from receiving the products to analyzing the data.
Alright, so these are my materials that we're going to be using to do the MitoPlate S1. First, we have the plate in its packaging. Here we have the dye mix MC, which is at 6 times the concentration. We have our Biolog mitochondrial assay solution, which is at 2 times concentration. And I've also diluted it down to 1 times concentration with 1 part water and 1 part this solution. Here I have my stock solution of saponin, which I have at 20 milligrams per milliliter. Here we have the trypsin that I'll be using to remove my cells from the flask. I have sterile water. I have a 12-channel micropipette and 2 handheld pipettes at 20 microliters capability and 200 microliter capability. So here I've calculated out our solution that we need to put at 30 microliters per well into our MitoPlate. These are all at stock concentrations, but when we calculate it out, it'll all equal 1x concentration. So we're going to be adding our BMAS at 2 milliliters, our DiMC at 1.33 milliliters, our saponin 20 microliters, and sterile water at 0.66 milliliters, and that'll get us to 4 milliliters. I need 3 milliliters in order to pipette my solution into the wells, but I'm going to leave 1 milliliter for extra pipetting room. Now once I have this solution dispensed at 30 microliters per well into the microplate, I'm going to be incubating the plate for an hour to allow the substrates to dissolve. After that time, I'll get my cells ready and I'll dispense them in 1x BMAS at 30 microliters per well so that there's 60 microliters per well total in the plate.
Kyle has created his own technique for pipetting out the right volumes while still accounting for pipetting error. We would remind customers that in the MitoPlate IFU, we have suggested volumes that customers can refer to when preparing solutions.
So now I'm going to prepare the solution. These are our Biolog reagent reservoirs. First I'm going to add our BMAS. Next I will add the dye mix MC. Alright, and here we have our saponin, and this is going to bind to the cholesterol groups in our cell membranes, and it is going to create pores to allow the substrates and the dye and everything else to get in. One thing it's not going to do is create pores in the mitochondria because they're very low in cholesterol content.
A note on saponin. According to the instructions for use, we recommend that our customers use a final concentration of saponin between 12.5 micrograms per milliliter and 100 micrograms per milliliter. This range is left intentionally wide to account for different testing conditions. We've done a lot of testing with different saponin products in the MitoPlate assay and concluded that one product in particular is best. That product is Sigma Aldrich's saponin, product number SAE0073, which has some of the highest content of sapogenin of any product available, between 20 and 35% in any given stock. We've seen that this product has a very forgiving range of effective concentrations, which we attribute to the high sapogenin content. This means that our customers will see more reliable signal in our assay using this product. Therefore, we highly recommend that our customers use Sigma's SAE0073 product for running our protocol.
Next, I'll add my water. And this is just to get the final concentrations at all the right amounts. And then I'm just going to pipette up and down to mix everything around and distribute the solution evenly.
Some sections of the video are sped up to save you time, but we would remind customers to take care when pipetting and dispensing solutions to minimize bubbles.
Now that I have my solution ready to go, I'll get the plate out. And like I said, for this part, I'm going to be doing 30 microliters per well. My multi-channel pipette.
Kyle is using the same programmable 12-channel pipette from Vista Labs that we provide with our PMM systems.
Alright, and I'm going to gently tap down to make sure that all the liquid is at the bottom so that our substrates dissolve properly. And next, I'm going to incubate it for an hour. So these are my colon cancer cells, which I've been growing for about 2 days. They're pretty adherent to the flask, so what I'm going to need to do is remove the medium. I'm going to wash them with PBS buffer solution, and I'm going to add trypsin to remove them from the flask. Then I will resuspend them in BMAS after centrifugation. So I will be transferring the old media to 15 ml conical tube. And now I'll be washing them with Dolbeco's phosphate buffered saline. And this is approximately 10 mils just to get all of the old media out of the flask. And now I'm aspirating the saline solution and getting ready to put the trypsin in. And for a small flask like this, which is 75 centimeters squared, I'm going to be using 2 milliliters of trypsin. And I want to make sure that all the cells are covered evenly with the trypsin. And now I'll incubate this for approximately 4 minutes. And I'll know it's ready when all the cells are free-flowing and off of the flask. So now that we have our cells free-flowing and they've been trypsinized, we're going to deactivate the trypsin with our old media. We're going to take about 2 mils of it. And we're just going to wash up and down on the flask. And we're collecting our cells at the same time that we're deactivating the trypsin. Now I've removed all the cells and I'm going to put it back into the old media and get ready to centrifuge it. Alright. Now we can see all of our cells are down here at the bottom in the pellet. So we're going to aspirate the supernatant and then we're going to resuspend it in our 1x BMAS solution. So I'm going to resuspend it in my 1x BMAS solution. And I'm just going to do around 4 to 5 mils prior to counting. So now I have my solution of cells in BMAS, and I'm going to dispense it through a 70 micrometer cell strainer. And what that's going to do is that's going to evenly distribute the cells throughout the solution, and I'm going to remove the clumps of cells so that it's easier to count. There's a number of ways to count cells, but the method that we're going to be using today is a trypan blue exclusion. And so I'm going to take 80 microliters of my cell suspension in BMAS. And now I'm going to add 20 microliters of trypan blue. So now I've calculated my dilution to have 1,000,000 cells per mil, which is going to yield my 30,000 cells per well. So I'll be diluting it in my 1x BMAS buffer. And again, I'm going to pipette up and down to uniformly distribute my cells. This is going to ensure that the correct number of cells gets into every well. Alright, and now that I have my cell suspension ready, I'll go and get my plate, which has been incubating at an hour. So now all the substrates are dissolved in the solution, and we can add our cells and get ready to put it into the Omnilog. And I'm going to dispense at 30 microliters per well. Okay, and again I'm going to lightly tap at the bottom just to make sure that all liquid is in the wells. And now we are ready to put it into the Omnilog. So for the MitoPlate I-1, we're going to do pretty much the same procedures with the same assay mix. We're going to add it to wells A1 to A4, and then after that we're going to add in either succinate or malate, and we're going to add that to all the other wells.
The MitoPlate I-1 protocol is very similar to that of the S-1, with the main difference being the use of 2 different assay mixes, one with and one without an additional substrate. This is accommodated by Kyle's technique of switching out pipette tips when adding the different substrates to the proper wells of the plate. After between 3 to 4 hours in the Omnilog, the data can be pulled and examined in our analysis software. This concludes Kyle's demonstration of the MitoPlate protocol. We observed the detailed steps at every point to ultimately examine mitochondrial metabolic function of a Colo 205 cell line, from receiving and preparing solutions, to plating the cells, to adding the plate to the Omnilog, and finally to analyzing the data. We hope that this video was not only informative, but that this product inspires you with new and unexplored ways of analyzing mitochondrial metabolism through a convenient and powerful avenue. If you would like to learn more about the potential applications of the MitoPlate or any of Biolog's products, you can reach out to us through our website on the Contact tab, or you can send us an email at sales@biolog.com.